Hello, ladies and gentlemen. No matter what time zone you are joining us from, good day and a very warm welcome to this Bayer LinkedIn Live Innovation Awards ceremony. I'm Melinda Crane, a journalist with long experience in the pharmaceutical and healthcare sector. And on behalf of our host at Bayer, it is my great honor to accompany you as moderator. As many of you know from experience, recent years have seen groundbreaking developments in genomic cancer testing and in next-generation sequencing. By significantly improving our understanding of tumors, these developments can markedly enhance treatment outcomes and quality of life for adult and pediatric patients with rare genomic alterations. So it is imperative to get the word out to ensure that these innovations in cancer care are available to all patients who need them. Patient groups and nonprofits are playing a crucial role in doing just that, in fostering public awareness and access, and the Precision Oncology Patient Innovation Awards, PIA for short, that we bestow here today for the second time recognize exactly those efforts. Joining us remotely is an outstanding group of experts. I'll introduce them in just a moment, but a quick word first on what's in store. After greetings from our host and a keynote from a renowned researcher who's devoted her career to developing personalized therapy and treatments for rare cancers, two distinguished members of the POPIA jury will introduce this year's winning projects. Following the winners' short presentations, a panel discussion will explore barriers to access and how we can overcome them. Dear ladies and gentlemen, we want to make this discussion as lively and interactive as possible. You can share your questions and your brief comments using the chat function on our live stream. And now, it is a great pleasure to introduce our guests, and I'll keep the intros brief to maximize time. Dr. Abdel Ali Majdi is head of Global Medical Affairs Precision Oncology at Bayer. Dr. Marcia Brose is professor of medical oncology, director of cancer services, and vice chair of hematology oncology at the Sidney Kimmel Cancer Center Northeast, Jefferson University. Scott Howard is professor at the University of Tennessee Health Science Center, secretary general of the International Pediatric Oncology Society, CEOP, and CEO of the global informatics company Resonance Health. Dr. Benedict Westphal is head of the Molecular Diagnostics and Therapy Program and the Molecular Tumor Board at the University of Munich's Comprehensive Cancer Center. And we also have two further guests, as you can see, who will remain nameless for now. They are our particularly special guests, and you will meet them later on. And now, without further ado, let me hand over to our host, Ali Majdi, to tell us why this award is so very important for Bayer.
Thank you, Melinda. I'm very pleased to be here with you today and represent Team Bayer at this very special event for us to celebrate the winning projects for the Bayer Precision Oncology Patient Innovation Award. And also celebrate all the projects we received for this grant, all the people working continuously to support patients living with cancer globally. So I'm here to represent the Bayer team. I have the privilege to work with many brilliant colleagues around the globe, from early research to commercialization, really committed to bringing innovation to patients around the globe, working with amazing experts such as the ones we have with us today, scientific societies and patient organizations. In the last few years and for the years to come, a wave of innovation in patient-centric precision oncology is coming, shifting the cancer treatment from one-size-fits-all to more personalized, tailored treatments for individual patients. And we are moving from the location of tumors, talking about breast cancer or lung cancer, to what we call tumor-agnostic approaches to treatment, developing medicines to treat specific alterations in the tumors for individual patients. And these innovations open, of course, many opportunities, but also avenues for collaborations to further develop research areas like new targets, new study designs, and more importantly, how to make this innovation accessible to patients. And this is why we are here today, to support projects to foster access to this innovation globally. We are here today for you as a company leading precision oncology and as a trusted partner. Thank you for your trust and looking forward to continue this collaboration with you.
And Ali Majdi, thank you very much for that. We hear next from a keynote speaker who is passionate about improving access to genomic testing and precision oncology. Marcia Brose, you have the floor.
Thank you, Melinda, so much. And it's such a special thing to be here today. This is definitely the topic nearest and dearest to my heart. I will just give you a couple of slides to sort of talk about a little bit where we've been and where we're going. Can we go to the next slide? So I actually did medical school. I also did a PhD in genetics. So of course you were probably not surprised to find out that this would be something that I would love so much. But in 1995, if you go to the next slide, when I first started in medical school, it really turned out that most of the patients who had metastatic cancer at that time, it was really a death sentence and in not too long a period. So many of the people who had stage 4 disease could expect to maybe live a couple of years, few months, maybe, and they wouldn't do very well. And that was just not very, it wasn't very hopeful for most of those patients in that situation. And that was partly due to the lack of effectiveness of systemic therapies. The one size fits all many times was not enough to get the patients to where they needed to be to have their disease stabilized, and if anything, even to regress a little bit. So patients then faced either having to have no therapy at all and just go into hospice, or sometimes to have these systemic therapies that unfortunately would be very, very difficult to tolerate, very toxic, and then their last years or months would be left with severe toxicity, and in some cases, multiple hospitalizations. So let's go to the next slide. So then how do we get here? Well, around the same time in 2003, a little bit after I started my assistant professorship, the Human Genome Project was published. So that was the first time we actually had the gene sequence for every single gene in the human body. And this rapidly gave rise to what was called the Cancer Genome Atlas Project. And labs that I was working in at the time were part of this program, which really went and said, okay, we know what the normal human body, for the most part, and the variability that we would expect in normal tissue. What do we have in cancer tissues? And how can we actually make sense of that? So an effort was made to start sequencing the DNA of multiple cancers. And with that, we found that sometimes there were similarities and sometimes single alterations that were actually likely the cause of those cancers. And what was really striking was that for the first time, a cancer from totally different parts of the body might actually be caused by the same mutation. This really turned on our heads, the way we looked at cancer. We used to think of it really by systems, the GI cancers, thoracic cancers, skin cancers. Now we actually knew that behind all of that, there was the genetic code, and that sometimes that was probably the most important. And with the identification of those alterations came targets. Those were targets that then pharmaceutical companies could start to try to aim at in order to get a response to help develop new therapies. And so this was a lot less of a blind lead the blind kind of approach, but much more an awareness of what was causing the cancer, and let's go right to the cause. So let's go to the next slide. So why target gene alterations? And I'll just give you 2 examples from my experience. So the first one is the next one. Could you just hold the talking for a second? So the next is consider a patient. Okay, this was a mother of 2 with metastatic sarcoma. And she was in a wheelchair when I first met her, and she had been on high amounts of oxygen because her metastatic lung sarcoma filled her lungs, okay? In a wheelchair on oxygen, and was told basically 3 months to live at best, okay? Her tumor is sent for genetic testing, and it turns out a gene fusion is identified, and it turns out this is one of the ones that we have a targeted agent for. Within 2 weeks of being on the clinical trial, she's off of oxygen. 2 months, she's out of the wheelchair. And at 2 years, there's now no longer evidence of disease. So we've now taken the worst actor metastatic patient and treated them, and amazingly, not only gotten to the point where we could stabilize their disease, but now it's completely gone. Now let me go to another patient. So if we go to the next slide, I had a 50-year-old who had a metastatic carcinoma that was from the salivary gland or the thyroid, and it was wrapping around a blood vessel that was really important for life in the base of his skull. He was basically told, go home, get your affairs in order. You won't be around for 2 or even 3 months. Again, he came to my office. He had an abnormality that made me suspicious. We sent off genetic testing, which is something that I like to do on all of my patients. And it turns out he again has this gene fusion, goes on a clinical trial. Within a couple of years, now we can no longer find his cancer. Now, in this case, he had other medical issues, and after 5 years, he had to go off of the drug. And miraculously, his cancer has not come back. So now we're talking about not only controlling or completely eradicating evidence of the disease, but we might be getting into a situation where these targeted therapies are really producing cures and very good responses in places we never have seen before. We're now 8 years out. This guy is clearly still with us and living a good life. So I just want to say that this is why we do this. Now let's talk about next steps. Next steps is really, we don't want to miss any patient that we could have a response like this in. And so to do that, the next slide, the most important thing we can do is get widespread testing. For the most part, if we can find the alterations, most of the times we can get the drug to that patient. But if we don't know it's there, we can't give those medicines. We can't give those drugs. So because of the fact that these new class of highly targeted therapies that have unparalleled responses, we need to find all of these patients. And agents in this category, actually, for most cases, are very easily given. I'll point out that instead of IVs causing big, problems and low blood counts that might lead people to, life-threatening infections, many of these drugs are well tolerated, and many of them are oral. So these are really ones that patients can take and live a full life. But identification using genetic testing on tumor tissue, not just blood samples, but on tumor tissue, will many times provide the key and will provide us a way for finding these patients. And in so doing, improve the quality of life for our patients, and in many cases, extend their life, extend the length of their life as well. So let's go ahead to the next and last slide. So in order to address this first challenge, we need to expand genetic testing on all cancer samples. I differentiate that from blood samples because sometimes the blood samples doesn't have all of the information. And in order to do that, we also want to make sure that all people have access to these testing agents, because if patients don't have access to the testing for any reason whatsoever, that's going to be a problem. So it's not just that we have the test, is that we make it available to as many people as possible. And then we will also have to follow up with that, of course, by pursuing improving the availability of the treatments. But that will really come second. I really think that identifying the patients is the first and most important step. Then access to testing will continue to be the challenge. And in so doing, we will need to address that in any health system across the globe. Every health system is different, and making sure that patients have access and that we can get them the information is going to be really what will make us be able to fully grasp the amazing results that these therapies give. So I thank you very much for your attention. Thank you for inviting me. Happy to be part of the answer at the end. So let's go do it. Thank you.
Marcia Brose, thank you so much for that good news presentation. And now, the moment we have all been waiting for, dear ladies and gentlemen, our awards. And let me tell you, it was not an easy choice. Our jury, composed of independent experts with strong representation from patient advocates, as well as the medical community, reviewed dozens of submissions from teams in Argentina, Brazil, Italy, Portugal, Spain, Saudi Arabia, South Africa, and Australia, to name just a few of the 50 countries. 4 projects were nominated, 2 winners were selected. Here's a closer look. So as you saw there, we do have two winners. Both are represented here remotely. I will hand over now to jury member Scott Howard to tell us what makes our first Popia Award recipient so outstanding. Over to you, Scott.
Thank you, Melinda. So that was great to hear from you, Marcia, about some amazing new miraculously cured. Well, we say it's a miracle, but it's a miracle based on careful clinical trials over many decades. And now I want us to imagine together a future in which every child with cancer also has a complete genomic analysis of their cancer so that their treatment can be personalized to include only the most effective therapies. This is exactly what the team at the Vall d'Hebron Hospital in Barcelona, Spain, is working towards with their innovative project entitled PENCIL. And PENCIL stands for Personalized E-Medicine for Cancer in Children in Spain. So their aim is to write a new chapter in oncology by applying personalized medicine to every child with cancer in Spain. So I'm delighted to announce they will receive $45,000 from a generous grant from Bayer to support their work. And with us today is project team leader Aroa Soriano Fernandez to tell us more about it. She got her PhD and is a genomics researcher. Together with her pediatric oncology colleague Lucas Moreno, they lead the personalized medicine group of the Spanish Society of Pediatric Hematology and Oncology. So congratulations to the team at Vall d'Hebron, and over to you, Dr. Fernandez, to explain more about the project.
Hello, everybody. Thank you very much, Scott, and we are very happy to receive this award. And thanks, Bayer, for your commitment to personalized medicine and for supporting us in this project to implement the personalized medicine in pediatric cancer in Spain. Thank you very much. So I start with the presentation. Pediatric cancer is considered a rare disease. It's different from adult cancer and is still the first cause of death in children and adolescents in Spain. The Spanish Society of Pediatric Hematology and Oncology has recently analyzed the status of the personalized medicine in Spain and detected that only 40% of Spanish hospitals have access to sequencing panels at diagnosis, and 60% of them have access to these next-generation sequencing panels at the time of relapse, when identifying new therapeutic targets is critical. So the main objective of the PENCIL project is to facilitate access to precision oncology sequencing studies for all children and adolescents from all Spanish hospitals. We want to develop a comprehensive sequencing program, including next-generation sequencing panels at diagnosis for high-risk patients, methylation profiling for central nervous system tumors and sarcomas, and a deeper sequencing with whole exome sequencing and RNA sequencing at relapse, and also germline analysis. This personalized medicine program will allow us to improve diagnosis accuracy, increase treatment options with targeted therapies at relapse, and to identify cancer predisposition syndrome. Next slide, please. Thank you. The specific objectives of our proposal for the Bayer Precision Oncology Patient Innovation Award are to create a network of clinicians and genomic centers. In Spain, we have 50 hospitals treating children and adolescents in Spain. And in the PENCIL project, we have 10 genomic hubs. We want to create a network to facilitate the inclusion of patients, submission of samples, sequencing, the discussion of the genomic findings in national molecular tumor boards, and return the recommendations to treating physicians. And we also want to create, that this is very, very important also for us, procedures to harmonize standards in genomic testing and the interpretation of molecular results across the country. And our second objective is to promote dissemination activities to raise awareness of the need to incorporate personalized medicine in routine care in Spain, and also activities targeting families of children with cancer. In the end, we would like to increase the access of pediatric patients to personalized medicine genomic studies in Spain and achieve equity and excellence in care for Spanish children and adolescents with cancer. So thank you very much. Thank you for inviting us to this nice ceremony and for your support. Thank you.
Congratulations.
Indeed. Thank you, Aroa Soriano, and congratulations to you and all your team for that very exciting and hope-giving work that you are doing. Before we hear about the other exciting project singled out for a POPIA 2022 award, dear ladies and gentlemen, let me just remind you that after this, we will be taking your questions or short comments. So please do use the chat tab to send them to us, because we really do want to be very interactive and hear your thoughts and questions. And now, over to jury member Benedict Westphalen to tell us about our second award winner.
Yeah, dear colleagues, friends, and watchers and listeners worldwide, it's my great pleasure to announce not the second, but the co-winner of the Bayer Precision Oncology Awards 2020. And we as the jury felt strongly that worldwide access to innovative diagnostics is the key to push precision oncology forward. Secondly, it became quite apparent that we felt strongly about the potential to push precision oncology, especially in childhood cancers. And this is why we decided that the second project, awarded with $45,000 by the POPIA, the Bayer Precision Oncology Awards, goes to the COPPA initiative by, and please excuse my Spanish, the Foundation Natali Daphne Flexer from Argentina. And this initiative aims to allow access to comprehensive genomic profiling for pediatric and adolescent patients with cancer in Argentina. And what we felt very strongly about is that this initiative aims to build a network, not just aiming at university hospitals, but really bringing innovative diagnostics to patients wherever they live. And we will now hear about this great initiative being awarded by Dr. Andrea Lecha, who received her PhD from University of Buenos Aires and is the principal scientist for the National Council for Scientific and Technology Research. Andrea, over to you.
Thank you very much. And in the name of Fundación Flexer and the COPPA team, I thank you very much for considering our COPPA project for this prestigious award. I'm going to explain why we thought our proposal fit exactly with the spirit of the Precision Oncology Patient Innovation Award. Pediatric oncology, as Aroa already said, is one of the arms of oncology most in need of a precision medicine approach, as pediatric cancers are per se rare diseases with a genetic basis. Treatment of pediatric patients needs to take into account not only the healing aspect, but also the long-term quality of life after cancer, which is an endeavor because many struggle with the secondary effects of chemo or radiotherapy regimens. Argentina, as part of Latin America, is behind other regions in access to a personalized approach to health. And this is mostly because of the belief among healthcare providers that there are more urgent needs than costly treatments associated with precision medicine. And this is, in our opinion, a very limited view of what precision medicine is. In fact, many pediatric cancers still lack these expensive tailored therapies because of the genetic basis are very different from common adult tumors. And so The genetic characterization of pediatric tumors serves two equally essential needs: the search for better therapeutic approaches, as we mentioned before, and the recognition of new drug targets. This combination of rare and essential, these are the words that led us to propose the COPA project, which aims to provide genetic characterization for all high-risk pediatric tumors in Argentina at no cost to the patients. The challenges are many, but Fundación Flexer is not shy in accepting those challenges. Fundraising for sustainability and logistics to guarantee access, these are its aims. Fundación Flexer has already established a network all across the country to ensure the samples will get to the sequencing center as soon as possible. Oncologists and pathologists are essential in providing the cases and their clinical pathological characterization to aid in identifying the sample to process and make the correct interpretation out of results. They are committed people for all over the country who identify the patient and the best samples to send. The help of trained pathologists will allow a complementary analysis if necessary to establish a primary diagnosis. And we at the sequencing facility are tasked with processing each sample, sequencing it, and interpreting results in clinically compatible times using QC, QA practices and in an affordable way. We aim to use a commercial targeted panel that includes most actionable genomic features so we can focus our analysis on the most clinically useful information. We are also going to participate in international proficiency tests to have a good measure of our analytical validity. Each genomic finding will then be recorded in a customized resonance database, along with the most significant clinical and pathological characteristics, as the genomic landscape of pediatric tumors in Argentina constitutes a valuable piece of information for descriptive, prognostic, and predictive objectives. Finally, the struggle with the interpretation of genomic information of those professionals that must take decisions based on them usually plays against the adoption of a precision medicine approach. The COPA project promotes the discussion of findings in the context of a multidisciplinary tumor board, believing that this is the best way to foster, among all intervening professionals, understanding of the consequences genomic findings have in the prognosis of cancer and its response to treatment. We firmly believe that Project COPA is a game changer in our Latin American reality, and thank you very, very much again for believing in us.
Fascinating look at your initiative. Again, also offering hope to
Melinda, you are
children.
Mute. Congratulations, Andrea and her team.
Okay, let me see if my sound is on now. Yeah, I guess it is. Very good. Also to you, Andrea, congratulations, and thank you so much for that fascinating look at your initiative that gives so much hope to children in your country. Dear speakers, ladies and gentlemen.
While we're sorting this out, everybody, the question line is open. Please put your questions in the chat or your comments. And I really, really appreciate the three speakers.
Can I ask a question?
Let me ask whether
I was just wondering, for both of these projects, is there also a way, is there a lot of attention being made on how that information is getting back to the clinical record? Because sometimes I know getting the sample to the site is actually only half the equation. And in our site, sometimes finding that data, lost in the charts can be somewhat daunting. So I didn't know if that was something that you have thought about or already considered in your proposal. So, Andrea, since I was just listening to you, why don't you start? Do you have, in Argentina, is there a way to get that information back?
Well, we plan to give an informed paper-based inform or report of the findings as any other commercial platform that provides this kind of report. So that's the way that we thought we are going to go back with the results to the physicians and ultimately to the patients.
Thank you very much to Scott and to Marcia. Can you hear me now, everyone?
Yep.
Thank you so much for jumping in there. And we will now move straight away to our discussion. And dear ladies and gentlemen, to kick it off, we have a poll question to you, to the audience, because it's clear that to broaden access, the first step is simply to understand what we are talking about. Because the terms associated with precision oncology, tumor markers, genomic testing, and so on, certainly can be confusing. So please start by typing into the chat which answer to this question you think is right. Our question is, what is genomic testing? Please type A if you think it detects hereditary alterations in DNA, and B if you think it detects acquired alterations in DNA. And we'll wait just a moment for results, and I think I'm not giving anything away if I say that while that question sounds deceptively simple, it is not. And my own first shot at answering it was, in fact, wrong. So let me see if we now can get a look at some first results here. I'll ask our technical team to give me, nope, they are still coming in right now. So let us just move on to a quick round of exchange while we are waiting for those results to come in. And I don't see any questions coming in yet from the audience. So let me just ask all of our speakers to talk about where we are on the path toward precision oncology. Is the goal in sight, namely access for all? And what do you think are the main barriers to widening access, to reaching that goal? And let me start, first of all, with Scott, because I know you have done a lot of work on access in lower and middle income countries.
Yeah, thank you. So I would say we are still on the path. In fact, probably we're just taking the first couple of steps on the path. So in the case of children, it turns out only 15% of children live in high-income countries like US, Europe, Japan, Australia. 85% live in low and middle-income countries. So if we really want to cure every child with cancer, we can't just do Spain, we can't just do Argentina. It's got to be everybody by the end of the day. So that, for me, is the beauty of some of the new targeted therapies. Many of them are oral. Many of them have very nice toxicity compared to the old cytotoxic chemotherapy. So suddenly, places where you could never give high-dose chemotherapy, like sub-Saharan Africa, for example, what we've done is adapt the treatments down to decrease toxicity, but then people relapse too much. So you can't control the disease without the high doses of chemo, unless you have a targeted therapy. So getting those targeted therapies, not out of Europe and the US and Japan and Australia, but there too, but bringing them to the other places where they're needed. This is why I'm actually really happy with this project, both projects, because the idea is some people in Spain get genomic testing, some don't. How to get it to everyone in Spain, everyone in Argentina. That's a great step one. And then we can start working on other places. So I really appreciate both of you for your projects and most of all your vision to get this done.
Thank you so much, Scott. And let me go, in fact, to Aroa and Andrea to talk a little bit more about the challenges of coordinating care and raising awareness in your respective countries, because clearly coordination and collaboration are absolutely crucial here. Aroa talked about that in her presentation. So maybe you can say a little bit more about that. I'll start with Aroa, if I may.
Okay. We have many challenges to coordinate 50 hospitals and 10 genomic hubs. And the first thing that we want to do is to conduct a survey to update the needs of the hospitals and in relation with personalized medicine, and also to know exactly the technologies that are performing all the genomic hubs to organize this network. And we want to centralize and to create a unique point of call for all the clinicians from the 50 hospitals in Spain to organize this network.
Thank you very much. And over to Andrea to talk about it from your perspective.
Well, yeah, well, it's always the same. The logistics of this connection between all the different actors, it's quite difficult when you on a country-based. Fortunately, we have Fundación already solving these problems because all pediatric diagnostics have already this network working on for common pathology determinations. So this is an advantage that we can use in our COPA projects.
Thank you very much. And we have a question that has come in from the audience, and Scott has actually moved to answer it in the chat itself. But I'm just going to let you briefly say to the whole audience, Scott, what you have written to the questioner. The question was about the cost of precision oncology, especially for developing countries. So just a quick sentence on that, if you would, Scott.
Yeah, the cost of drugs starts actually upstream. Ben, you want to take this one?
No, go ahead, but I have comments on this as well, not just in developing countries.
Because the upstream cost is the cost to develop a new medicine, and that cost is way, way, way too high. So what we need is not just access of developed medicines to everyone in the world, but we also need access of everyone in the world to research studies so we can get these drugs out the door much faster. And in fact, especially middle-income countries, 75% of children live in middle-income countries. Argentina is an example of a middle-income country. In fact, most of Latin America are middle-income countries. These are countries that have research capacity, have terrific doctors and scientists, like we're hearing from Andrea today. So these are places where research can be done to try to get the drugs out the door faster, which means also it's cheaper, because if a study takes 5 years, it means it takes $200 million instead of only $100 million. So to get drugs cheaper, we have to make them cheaper, and then of course get them out the door. So I love your question, Ovenucci, because how do we get these drugs to certain countries? And the answer is donation programs, which is actually what Bayer and several other companies have developed. That's the short-term answer.
Thank you so much. And let me go right over to Ben. You wanted to speak to that point as well?
Yeah, and secondly, I mean, the question is, if you don't diagnose a patient who might actually qualify for targeted treatment, the best drug in the market is useless. And luckily, within the next 1 or 2 years, we're going to see a lot of patents run out that make good molecular pathological diagnostics so expensive at the moment. I mean, we're talking 4,000 or 5,000 euros, dollars, whatever you want to name it, for testing, which is compared to what a whole cancer journey for a patient costs, not a lot of money. But in the end, we're going to be able to provide the same technology for a fraction, namely 200, 500 euros or dollars. And that's a complete game changer, I believe. So the next couple of years, not only making drugs cheaper, but also making the diagnostics cheaper is going to be, seminal.
Thank you very much. This is essentially now another question that's come in from Rakchanda Kuchi, following up on what we heard there a moment ago from Scott in regard to the fact that there are excellent research facilities in so many middle-income countries now. But this questioner asks, how will technology be provided to places and countries where advanced research is not going on in the field of oncology? Do you also want to take a stab at that one, Ben?
Yeah, I mean, what I believe is that precision oncology can only, medicine can only thrive, maybe society can only thrive in the future if we cooperate across the globe. I mean, I just sat together with a good friend of mine last night. He's a lawyer, and there's so many unsolved questions that we face worldwide. Let it be climate. But coming back to today's topic, precision oncology can really serve as a catalyst to help us think medicine in a whole new way, talking about integrated healthcare solutions. The best drug is really useless if you don't find the patients that qualify for the drug. So in order to find these patients, you have to think about how can every patient suffering from cancer get access to high quality diagnostics. When you think about high quality diagnostics, you have to think about rolling this out in a quality ensured fashion. So even while we have drugs that only target a very, very small subset of patients, these new drugs, because they come on the heels of good diagnostics, have really the potential to change our healthcare system. And this can only be done if we work together, not only focusing on our ivory towers that we work on, just really working together on a global scale to help our patients.
Thank you very much. I want to go to Marcia, if I may, also with an audience question that has come in, and then a follow-up from me, if I may, on communication. And the audience question comes in again from Ovenuchi Ejohuo, who says, what modalities are in place to ensure that targeted drugs consider the genetics of people in developing countries? Will researchers be collecting genetic samples from these places?
So that's a great segue, probably back to the answers to the poll that I'm sure you're going to give us. But, you know, at the end of the day, there are two things we're talking about. We're talking about identifying the genetic alteration that caused the cancer. And you can imagine that that's on the backdrop of the genetic differences that might be across different communities. But at the end of the day, believe it or not, we're much more alike than we are different. And so when it comes to these drugs, the most important thing will be we will develop them and they will really target them to a cancer, but they're often, as was said before by Benedict, this is also something that is often done on global trials. And in a global trial, if there is a signal because one population does better or worse, we will often see it at that time. And so I think that we can feel pretty good about the drugs that we're talking about most of the time are really going to be great across all populations, but they are being studied. And then there often are follow-up clinical trials in different populations just to make sure. But that's a great question.
So let us indeed come back to the poll, because we didn't get we don't have a really representative sample for you scientists, but what we can say anecdotally, as it were, we have about 2/3 saying genomic testing was answer number A, and 1/3 of the respondents say it was B. And of course, B was the correct answer. So clearly there are awareness issues. As I mentioned, my own answer was incorrect. Marcia, how do we raise awareness and also overcome communication barriers to help patients and families discuss the testing and targeted treatment options?
Well, how and that we need to do it are both very important questions. I think that there just needs to be a way that we talk about basically cancer mutations differently than the ones we talk about, hereditary mutations. And it can get clouded because, of course, a lot of this research started with hereditary gene mutations that cause cancer. So there is even an a place where they're both merging. So I think we have to put in time the same way you would do in an advertising campaign, communications campaign. We really have to educate people that there are two kinds of mutations. They're the mutations that are in every cell of the body and therefore could be tested in the blood that are hereditary. And then there are the changes that happen just in the tumors. But interestingly, sometimes you can have both. But if you don't check the tumors, you might miss half of the ones that you could target with these drugs. And so many times I think maybe the answer for this purpose is to really start to encourage people to test all the tumors, because many times we'll find mutations in the tumors that are not present in the bloodstream. And I think that's where the education has to be really directed, and I would almost imagine like a campaign that says, have you had your tumor tested yet? Something like that, basically, so that everybody knows that as much as they know who like the Komen Foundation is for breast cancer research. Like that should just be a thing everybody says.
Thank you very, very much for that input. And our time is just about up, but I certainly want to go back to our host and Ali, hear what Bayer is doing to address the kind of barriers we are talking about and thereby improve access for all.
Yes, thank you all. I just wanted briefly to come to 3 points mentioned in this conversation. One is really raising awareness and taking the example of, I mean, today we are entering the Lung Cancer Awareness Month, and we are not there yet. Many patients still even in the US, developed markets, don't have access to genomic testing for lung. Lung is one of the areas where precision oncology is leading the way, but we are not there yet. So there is a call to action for all of us to collaborate to change this paradigm in developed market, but as mentioned earlier, globally. The cost of technology, I just, Marcia mentioned earlier the history of genomic testing, and if we remember the cost that was to do the first whole genome sequencing was billions. Now we are talking about hundreds of US dollars, and hopefully this will continue decreasing. But beyond that, our teams are working on evaluating new ways of testing or identifying patients with genomic alterations, such as digital pathology. So using machine learning and AI to really overcome this access challenges to genomic testing. And I can tell you, as I mentioned earlier, we have some really bright people working within our companies and in collaboration with academia to really develop these ways of getting the genomic testing via the different channels. The last is really I like the question about understanding the impact of genetics on the performance of these targeted treatments. And here again, being open and for our trials to include more diverse population around the globe will help us understand how these treatments are performing in terms of effectiveness, but also safety in some populations. And the use of real-world evidence is probably another way we are all exploring now to learn more about our drugs and to help more patients globally. Again, I would like to thank our panel for their presence with us today and the 200 plus participants to this call. And back to you, Melinda.
And thank you so very much for that, Ali, and to Bayer as well for making this very valuable discussion, this very beautiful Innovation Award ceremony possible. This does wrap up our LinkedIn Live Innovation Award ceremony hosted by Bayer Precision Oncology. Many thanks to our speakers for the insightful discussion on the exciting role of genomics in developing more targeted, effective cancer care that does improve both outcomes and quality of life. And also for your insights on the crucial importance of widening access to genomic testing and precision treatment. All of you, as well as the dozens of teams who submitted candidate projects to this year's award, are working to deepen awareness and access, and we wish you the utmost success going forward. Many thanks to the 4 nominated projects, and once again, congratulations and thanks to our winners. And ladies and gentlemen, we are also very grateful for your attention and your contributions. In case you missed something or you'd like to hear our discussion again, you will find a recording on YouTube. We're going to be posting the link on our LinkedIn event page. Many thanks to Bayer for making the ceremony possible. I wish everyone a good afternoon or evening. Stay healthy, and goodbye.
Take care, everybody. Thank you.